Hey everyone, we're going to do notebook page 10, senses. This is the sensation and perception unit. So we're going to talk about how vision works, how audition or hearing works. So take down the title card now at this time. So let's start with how vision works. Sight is a fairly simple process, simple anatomical functions. And the keys to understanding is that the rods in the eye sense grayscale and the cones sense color. That's the difference between those. Most of our cones are located where light is focused in an area on the retina called the fovea, while rods become more populous on the extended part of the retina, kind of the peripheral, if you will. And the human retina contains about 6 million cones and 120 million rods right outside of it. So a lot of small pieces working in tandem here. Rods and cones send neurochemical signals to the brain through bipolar and ganglion cells to your thalamus via the optic nerve. And that's kind of how they work. We'll take a look at an image in a second, but I'll have you write this down now. Move on. So here's kind of a picture to get a visual representation of how this works. Here's kind of the parts that you've probably studied before: the pupil, the iris, the lens, etc. Right? We see the optic nerve here, as I talked about before. Right? That's what goes to your thalamus. That's where those neurochemical signals are moving toward. And then here's kind of how vision works. In terms of a picture, right? Light comes into the eye, and we see, this kind of photochemical reaction in the rods and the cones in the back of that retina happening as the light goes in. We see cones, we see rods, and we see them kind of processing this information. So it goes out the optic nerve, and that information comes in. So the other features. We have feature receptors in the occipital lobe that give us information on shape, on edges, on angles, to other parts of the brain so that we can recognize faces based on these things. Right? We talked about feature receptors before, how you can see faces and recognize them, and this is how that happens through vision. The occipital lobe kind of gives us that information. So this allows us to perceive perceptual constancy, recognizing things from different angles or colors for what they are. So that's so you can essentially recognize a person or an object, right, if they're turned a different way. If I see someone's face straight on, I don't have to see it straight on the next time to know who it is. That's those feature receptors doing their work. If I see a car that's red, the next time I see the same kind of car, it's green, I can still recognize it's the same type of car, even though the color is different. So, we can perceive all these at once. That's called parallel processing, kind of bring these elements in with our vision. So I'm going to put the slide up. You can write down your own words. And we'll move on to theories on color. So color is kind of a really interesting thing because we don't fully understand how and why we see color. Transduction is the alternating of one form of energy into another. As light energy activates the rods and the cones, it's converted into electrochemical energy when those rod cone receptors are active. They're activated. The brain can't see and detect light, so it has to go through that earlier process that we talked about at the beginning, through the bipolar, the ganglion cells, because the brain can't detect light. We have to convert that information. So how do we see color? How do we interpret color? We don't exactly know. There's kind of two theories, and neither are perfect. We have the trichromatic theory, that our brain sees colors and combinations mixing of red, green, and blue receptors. So kind of those RGB receptors, and depending on the wavelength, the color source, we know it's where it is within that red, green, blue kind of role, right? So, that's kind of what we have here on the right. We have different shades of these colors, and they kind of move through as they go. That we can kind of see those. So basically, using wavelengths and looking at this through different waves that are not visible to the eye, we can see color being visible in that way. So these three are kind of mixed by the activation of more of a particular type of color than the others, and it results in our perception. But this doesn't account for everything. Because if we look at a particular color and then our brain will actually see the opposite of that color. When we look away, we can actually see that. And I'll take you through this process after the notes. This is the opponent process theory, that we see colors through deactivation of opposing colors rather than seeing the real colors. And so, if you ever heard like the color black is the absence of all other colors or things like that, that's opponent process theory. So, we'll do some example after the notes of that. So you can kind of take these down now. All right, let's get into audition and hearing. Audition is comprehending of vibrations of energy in the air, which is sound. Vibrations occur in molecules that compose the air or water, and that surround us and are picked up by our outer ear. From there, vibrations will travel down to the middle ear, to the eardrum, which vibrates the hammer, anvil, and stirrup. And the stirrup transfers that energy into the cochlea, which is part of the inner ear. And those cochlear fluids are jostled to stimulate hairs on the basilar membrane. So this is kind of how hearing works. I'll show you an image after the slide here. So let me finish talking about it first. When those hairs kind of vibrate in the inner ear, they transduce the vibrations. They transduce the energy to neurochemicals by activating an axon, and sends that signal to the auditory nerve, to the thalamus, and then the auditory cortex, where it processes the sound itself. Process itself is pretty simple, but the key for our class is to note that the process of transduction, as various forms of energy are changed into neurochemicals for the brain to receive and interpret. Just like vision, the brain can't interpret light, but the brain can't interpret sound. So these, our eyes, our ears, our vision, and our audition have to convert that to neurochemicals that our brain can read, and that's how we get that sensation. So, these vibrations, these light waves are undetectable to the brain, so it relies on the activation of receptors that transmit the neurochemical signatures for identification and processing. So hopefully that makes some sense. We evolved to have two ears and two eyes to interpret a 3D world around us, so that we can determine distance. We can determine blockage if we see things, that are partially covered, that we know what they are, things like that. All right. So here's the picture I was talking about of the ear. There's the outer ear. It'll go through this into the eardrum, and then we see these small bones work, boom, 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 work together, and the stirrup will make that vibration, and that's what hits those kind of hairs, if you will, makes those hairs vibrate, and that's how we get the sound of what different sounds are. Now, just like vision, we don't fully understand how sounds work. So, we're able to interpret sound variants in different ways. Most notably pitch, which is kind of the frequency, and loudness, which is the amplitude, right? How loud something is. With the latter being measured in decibels, hopefully something you've had before, 10-fold increases in amplitude for every 10 decibels. We can measure loudness and sound. We'll talk more about that. Now, for loudness, only certain hairs are stimulated by specific amplitudes. So the louder something is, the more hairs that are affected. So that's how we can tell how loud something is. However, for pitch, it's kind of less clear. The theories that explain how to interpret pitch, there's kind of two, just like color. we have place theory and frequency theory. Place theory holds that hairs in specific places are stimulated only by certain frequencies, much like how we determine loudness, right? So it could be very similar, but there's some evidence to suggest frequency theory that, which assets that our neurons match the frequency of the vibrations in our brain, is able to interpret the frequency of that neuron signal. So it might not be the hairs, but it might be how our brain interprets the pitch that actually makes the pitch. So kind of interesting that we don't fully understand that, and that there's some evidence both ways. All right, we got one more here for sensory disorders, and this is something that we may have talked about a little bit before we did the notes. But one can lose their audition or vision, or hearing due to mechanical or neurological failure. So it could be an issue with your eyes or your ears, or it could be an issue with your brain. If the problem lies with the receptors or any related neurons, the optic nerve, the auditory nerve, parts that are neuron brain based, one would suffer from a sensory neural hearing loss, or with vision blindness or color blindness. So, blindness can also occur mechanically. It can occur due to damage of the eyeball, that light's distorted or light's blocked. Deafness can occur mechanically if the eardrum is damaged. So this is known as conductive hearing loss, and a cochlear implant can provide alternatives for patients that to hear by mimicking the eardrum and its parts. But if you lose out on the sensory neural piece, if it's the nerves, if it's the neurons, then you can't hear. And hearing loss is almost always permanent. We don't have a way to replicate the neural activity, just the mechanical activity. So, even with cochlear implants, it really, the effectiveness depends on the why and what the problem is. So you can go ahead and take this down. And for the other things on here, the synesthesia and color blindness, I probably will talk about in class. And that is all for this notebook page for sensation and perception. Thank you so much, and have a great day.